We'll get started. Everyone, welcome to the AOS Student Research Grant Seminar Series. Welcome to our final week. This is the last of what has been 15 presentations that I hope people have checked out or enjoyed here in the spring semester. I wanted to just say I'm Robert Driver. I'm a postdoc at Duke University. And as you know, from each week that I've introduced this, the AOS Student Research Grants are offered each year to early career researchers. And the early career researchers have to write a proposal that advances ornithology while still remaining feasible within the budget. And so that creates a challenge for AOS members, and we want to celebrate everyone who won that with their innovative work by sharing that work as part of the AOS Student Research Grant Seminar Series, which we're at the final one here today. And I just wanted to just add another reminder that we will return in the fall for another, what should be another 15-week seminar series starting just a little bit after the AOS meeting in late August. So before we turn our attention to Elkin's research, Elkin Tenorio, our speaker today, I just wanted to remind everyone to keep your microphones on mute. We will have a Q&A session at the end of Elkin's talk, and you can participate by writing your message in the chat or by writing in the Q&A box Zoom feature at the bottom, or by raising your hand, which is under the React button at the bottom, and I can unmute you and you can then speak your question. And I'd like to remind everyone that this is an AOS-sponsored event and follows the AOS Virtual Meeting Event Code of Professional Conduct, and I'll post a link to that in the chat. I'll also post a link to our post-seminar evaluation form. It would be great if you could take a moment to fill that out. It would really help us improve the seminar as we move into the fall series, which we'll have in a few months. And I'll also post a link to contact the AOS Student Affairs Committee, which helps support the seminar series. Okay, so our speaker today, Elkin Tenorio Moreno, is currently a Ph.D. candidate in biological sciences at the City University of New York. Not for long, though. It sounds like his dissertation is just around the corner. And Elkin's academic career began with a Bachelor's of Science in Biology from Universidad de Valle in Cali, Colombia. And Elkin then went on to earn his master's degree in biological sciences from Universidad de los Andes in Bogota, studying how climatic niche breath influences avian diversification, which sounds fascinating. Following graduation, Elkin worked in a variety of research positions, including 4 years as a research assistant at the Alexander von Humboldt Institute for research on biological sciences. And in 2018, Elkin was awarded a competitive Fulbright grant. And in 2019, Elkin began his PhD in the lab of Dr. Brian Smith at the American Museum of Natural History. And Elkin has made substantial contributions to Colombian ornithological research, with his work published in Proceedings of the Royal Society B, Global Ecology and Biogeography, BMC Evolutionary Biology, Ecology, Conservation Biology, Journal of Biogeography, among others. And in addition to the AOS Student Research Grant, Elkin has won the Lewis and Clark Fund Grant, the Society of Systematic Biologists Graduate Student Research Award, and the Fulbright, which I already mentioned. So with that, I will turn it over to Elkin for his talk, Origin and Maintenance of Andean Bird Fauna.
Okay, thank you, Robert. I'm going to start, I want to present this in this presentation, of course, thank you very much for people that attended my presentation. In this slide, I want to present like all my dissertation in which I was exploring like the ecological and evolutionary mechanisms that explain the high diversity in tropical Andes. So I want to start by showing this picture that just represent like on typical cloud forests in tropical regions. This is particularly from Chiapas, from Mexico. And I show this because it reminds me like one of the places that when I was starting birding in Colombia, I just fell in love of the all the diversity, all of birds, or the variety of the species and the colors that you can see in this kind of forest. And associated with that complexity that you can observe in the picture. But this complexity, if you zoom out of the picture, where you can see that this cloud forest is part of a whole gradient of other complex ecosystem that actually harbor astonishing levels of species richness. And I was very interested in how this, since then I have been very interested in which are the evolutionary forces that are driving all this diversity in tropical mountains. So what I have done previously is like I have tried to explore like general patterns of diversity across the globe. Like you can see in this map, this is a species richness map of the like a typical species richness in which you can see the number of species represented by, for example, red colors, a high number of species, and blue colors like low number of species. And what you can see here is like the normal, for example, latitudinal diversity gradient, right? Like the higher accumulation of species in tropical latitudes. But other patterns that you can observe here that probably are less obvious is like the high concordance of high diversity associated to mountain regions. So you can see the lines that are drawing here, like limiting the main mountain system around the world. And what you can see is that several of the main diversity hotspots are associated with those mountain ranges. For example, the Andean foothill, the Great Rift Valley in Africa, the Henduan Mountains in China, and the mountains in the Pacific Island. We have tried to quantify the diversity. Actually, we did that, for example, here for terrestrial vertebrates globally. And something that we found is that mountains represent like the 40% of the total diversity of the terrestrial vertebrates in comparison to lowlands. However, probably less people know that some people don't know that mountains just represent the 30% of the global land mass, right? So if we correct this number of species by the area that mountains occupy, actually we found that mountains can have until 2 times of more diversity per unit area than lowlands. This is even more impressive when we explore the same pattern of species per unit area. This is like a kind of measure of species packing, in which we have mountain here in red and lowlands in blue, and you can see the same measure across latitude. And what you can observe here is that there is almost an exponential increment of the number of species per unit area in mountains, particularly in mountains, reaching values up to 4 times more species per unit area in mountains than in lowlands. So why, yeah, how mountains particularly in the tropics can harbor these astonishing levels of diversity is probably represented by this picture that show what is a typical profile of a mountain in the tropics. And you can see there is a strong elevational stratification of habitats and environment that replacing even in very short distance. Species adapt to those particular environments. So if the environment change very fast, so the community of species also change very, very fast. And that explains this segregation of habitats and species. It can explain how we can put more species in less area. Actually, this is called like species turnover or beta diversity, right? And we also we have calculated this for vertebrates. And what you can observe here is that, again, high beta diversity is very high in the tropics, particularly, for example, in the Himalayas, that is kind of subtropic, but it's particularly highest in the Andes, right? More close to the Ecuador. So the general question that I wanted to address in my presentation was, which are the ecological and evolutionary processes that generate the high levels of diversity in tropical mountains? And these kind of complex questions need like a theoretical framework that include like processes and mechanisms that operate at different spatial and temporal scales. Like this we propose here, like including macroevolutionary processes, ecological mechanisms, and microevolutionary processes to understand the origin and maintenance of diversity in these regions. But this implies like also challenges in terms of limitation, computational, theoretical, methodological, that we hope we can address in the results that we are presenting today. So the first question at the macroecological level is, how is this diversity maintained while accumulating across time? So there are different ways in which species can accumulate. This is like a more theoretical way that is more like a linear, constant accumulation of the number of species across time, right? This is just constant. This is more theoretical. This assumes like more just speciation and not extinction. But with that theoretical model, we can estimate, for example, deviation from that model. And there are other patterns, like, for example, acceleration toward the present, in which, for several reasons, because new ecological zones or because new key innovation species, clades can accelerate the diversification, the number of species toward the present. But there is another pattern that is interesting for us, and probably most of the clades may follow this kind of pattern. It's more like a saturation model, in which the species increase early, very fast early in the history of the clade, but reaching the present, there is a saturation point in which the number of species become constant across the time. And this assumes like a saturation, some ecological factors that are limiting the number of species. And when we observe this pattern, we say that, okay, the diversity is in equilibrium. An equilibrium means essentially a balance between speciation and extinction. So actually, MacArthur have mentioned something about that. MacArthur says something that if some areas are not saturated with the species, an explanation based on diversification rates or a time for speciation will be appropriate. So he was referring to historical factors or something that we call also equilibrium. Sorry, not non-equilibrium. But he also said, if the areas are saturated with species, so the explanation of the number of species in a region should be in terms of niche space and limiting similarity. And he was referring to ecological factors or equilibrium processes. So we integrate or we try to synthesize the MacArthur idea in just one framework that I'm going to explain right now. So just to mention that I'm finishing with this theoretical explanation, but bits are coming in the presentation in a couple of minutes. So the diversity in a region just increased by the direct effect of historical factors like diversification rates and time for speciation. But this historical effect could act through the effect of ecological factors like morphological divergence. So, for example, in a region that there are more diversity because they are accumulating faster, species can coexist just because they can diverge in the phenotype in a way they can use different resources and reduce competition. Now, this reduction of competition could operate also through the species segregation. So species just colonize like different areas, so they are not together, similar species are not together, they are not using the same space, not using the same resources, and they reduce competition too. So in this case, this is the whole framework from which we build our predictions. So what we predict is that in equilibrium scenarios, historical factors have a positive effect over ecological factors, in this case, for example, morphological divergence. But, and more important, that ecological factors have also a positive and indirect effect over number of species. And that suggests that ecology is important and there is a compensation of ecological mechanisms by these ecological factors. But the compensation could be also through the effect of species segregation, right? But in non-equilibrium scenarios, what we expect is a negative effect of ecological factors over number of species because ecology is not important, should be not that important, so there is not a compensation of ecological interactions there. So the first thing that we did was to try to map which were the main radiation or mountain beat radiations, right? So we map like the minimal elevational the minimal elevation for a species globally, and we can identify like there are multiple beat radiation that correspond to mountain clades. But we focus in this case to three main clades that represent in this case tyranny, the subordinate tyranny, the 9 primary and the hummingbirds. They together have correspond to the 50% of the total neotropical avifauna. Yeah, it's about 2,400 species. But more important here is that these three clades have very good phylogenies over which we can make inference about macroevolutionary inference with a high confidence. So for all these species, we download a species distribution map, and the first thing that we did here was try to map the spatial patterns of species richness, and we essentially made like a stack of rasters. So we just put all the rasters together, and we sum all the rasters over pixels, and we can make a sum of how many species fall in the same pixel, in the same community. So what we obtain at the end is a richness map like this. Over this richness map, we quantify evolutionary factors. First, we quantify, for example, using the phylogenies of this clade. So we quantify, for example, diversification rates using different measures. Here I'm presenting, for example, the DR statistics, a measure that is very similar to the equal split. And we quantify, for time for speciation, we quantify the average community age. All these values, all these estimation over the pixel over the pixel of this richness map. For ecological factor, we estimate morphological disparity using, for example, a public database of morphological measure, ABONET, in which we use multivariate metrics to estimate, for example, in a community, how is the multivariate volume of the traits that the species that are together in a community. So the distance of this volume, of this morphological volume, could be small or could be big. For species segregation, we estimate beta diversity, but in this case, a measure of phylo-beta diversity, that is the same species segregation measure, but taking into account phylogenetic information. So what I'm presenting here is a little overwhelming, but I'm going to explain very, very slowly. But just to mention that we use structural equation models that we can measure, like, for example, different kind of interaction among ecological and historical factors. But over those structural equation models, we can infer non-equilibrium or equilibrium models for those clades. So I'm going to the more important relationships in these structural equation models, in this path analysis. And starting by hummingbirds, as you could remember, we essentially conclude that if there is a negative relationship between morphological disparity and species richness, that suggests non-equilibrium. I'm going to show this, I'm going to explain this more spatially in the next slide. But just to mention that tyranny also follow like a non-equilibrium model, just because, but through a different path in these models, through the effect of spatial segregation. And the only clade that follow an equilibrium model, and this means that ecology is important, limiting the number of species in those clades in mountain regions, was for oceans, right? In which there is a positive effect of diversification rates over morphological disparity, and in turn, morphological disparity over species range. Okay, I'm going to make this more clear just by specializing this in the elevational gradient. So what you can see here in this panel is that the diversification rate decrease along the elevation. So the diversification rate is higher at lower elevations. And that means that in this elevation, the species are accumulating faster, but in these same lowland regions, the morphological disparity is small. And that means that even species are accumulating faster in hummingbirds. In the lowlands, they are similar in the morphology, suggesting that ecological interaction probably in those similar species are not limiting the accumulation of species. So for the reason, in this case, we say there is no ecological compensation and there is non-equilibrium dynamic here. In the case of tyranny, we have an opposite effect of the diversification rate, in which we observe like a positive trend of diversification rate toward higher elevations. And that means that in higher elevation, species are accumulating faster, but in higher elevation, again, there is not a compensation, there is no particular increment in the morphological volume of the community, of the species in those communities in higher elevation. So it doesn't matter if that are accumulating faster in higher elevation, because probably they are not competing for some reason. But in the case of oceans, the 9 primary oceans, what we have is a negative relationship between diversification rates with elevation. So the diversification rates are higher in the lowlands, where the morphological disparity is higher. So that meaning that in areas where species are accumulating faster, there is an expansion of the morphological volume that probably is reducing ecological interaction in that region. So actually, equilibrium dynamics have been also observed in, for example, in other, in Himalaya, for example, works for Trevor Price. So to conclude this part of the presentation, what we can say is that even historical and ecological factors interact to facilitate the maintenance of the species richness across time in macroevolutionary level. Regional diversity may or not be constrained by those ecological factors, and that these three clades or multiple clades will accumulate diversity following different ecoevolutionary paths. Now, we want to, in the next part of the presentation, we want to dig deeper in the ecological factors and the important in the ecological factors, but in how ecological interaction operate in more recent temporal scales. So, for example, in a local scales, you can imagine a scenario in where there are several species, or imagine two species, that they are pretty similar in their ecology, the morphology and their ecology. So they are competing because they are using the same resources. So a potential result, there are two potential results in these cases where those species are competing. One is character displacement. So the morphology or the phenotype of the species or the ecology of the species diverge in a way that they are able to use different resources in a way that they can avoid the competition for the same resources. An alternative result, an alternative scenario is that one of the species is more strong in those competitive interaction, and that species exclude the another one to another place. And also, there is also a reduction of the competition because now they are in different places using the same resources but not together. So these two cases of ecological mechanisms are important. Have been quantified, like shown in model systems like Darwin finches or anoles, particularly in island regions. But our question is if those same ecological mechanisms that operate at local scale can also operate at regional scale, like, for example, the elevational gradients of tropical mountains, because species here have a strong elevational replacement that may suggest ecological interactions. For example, in the elevational gradient, you have species that occupy a portion of the distribution like this, like a narrow distribution in the elevational gradient. And this species is replaced for another species with a small elevational overlap. But this species in turn can be replaced for another species, but with a broader elevational overlap. So the question here is, what can determine the elevational overlap of those species? There is a case in a study of Jared Diamond in New Guinea, in which he was studying he studied many bears dead, but he exemplified like a case of competition by showing that along the elevational gradient in this mountain range, there were two species that they were increasing their abundance to this point of the elevation, right? But they when met together, probably the competition was so strong that the abundance of those species dropped dramatically in that point. So you think that the distribution of one species is limiting the distribution of the another one. And this is something that has been shown in not just in these two species, in multiple species across multiple tropical ranges. So for example, this study of this author show how species that are allopatric, meaning that they occupy different mountain ranges, they don't diverge in the elevational band. So because they are allopatric, they can occupy the same elevational band. So there is a low elevational divergence. That contrasts with the species that are in sympatry, species that occupy the same mountain range. When they occupy the same mountain range, one of the species occupy one elevational band and another elevational band, probably avoiding each other. So the elevational divergence is higher in sympatry. So the question that we have, the specific question that we have in this part of the in this component of the presentation was if interspecific competition is driving the elevational replacement pattern, and which is the role of phenotypes mediating competitive interactions in mountains. So the hypothesis that we have for this idea is that the proportion of elevational overlap predict the level of phenotypic divergence as a signal of interspecific competition. I'm going to explain this hypothesis here. Imagine that we have two species that are very similar in their phenotypes. In this case, color. They're similar in morphology and they're very similar in color, right? And because they are pretty similar, they try to avoid to each other in a way that they reduce competition. That is what we predict. So we should observe some in this relationship of the phenotypic distance and elevational overlap, we are predicting low levels of elevational overlap when there are low levels of phenotypic distance. But we have another case. Imagine two species that they are different in coloration in this case. So because they are different, they can coexist together. So what we expect is that when the phenotype is higher, the elevational overlap is also higher. And if we extrapolate this idea to many other species, what we have is like a positive relationship between phenotypic distance and elevational overlap. So the study system that we chose for this idea was the subordinate tyranny and oceans, like very diverse clades with very diverse family like Fornaridae, Tyrannidae, Grallaridae, Traupidae, Troglodytidae. But something that you can see here is also like a contrast in how colorful they are, right? Tyranny tend to be, there are some exceptions, especially Cotingas, but in Tyranny subordinate, they tend to be a little more uh, no, contrary. Oceans tend to be like more colorful than tyranny species. And there is an idea of what this could happen. And this idea is because of the habitats that they occupy. The oceans are species that tend to occupy the canopy of the forest, where there is more exposure to the light. And because there are more exposure to the light, probably they are evolving more colorful plumage because they are exposed, their coloration can be exceeded to another individuals. Probably the coloration is evolving to a more vivid coloration. But it doesn't happen in tyranny where most of the species tend to occupy the understory. Yes, there are exceptions. For example, tyranny tend to occupy the canopy, but most of the tyranny tend to be in the understory, where the environments are very dark and there is an scare UV, for example. So they tend to evolve dual coloration because probably they don't need vivid coloration in habitats where the coloration they cannot see the coloration of other individuals. So probably they are using another mechanism of communication, like for example the acoustic communication channel. So we expect like some kind of contrasting results in these two clades for this reason. So what we did to test this idea is that we look for phylogenies, we identify sister species, we identify the species that from those sister species, at least one species were in montane. And when, okay, after that, we quantify the elevational overlap among those species. We select the individuals for those species, and we use digital photography to quantify the color, and we also use morphological information for those same species. So as you may note, birds and humans we see in a different way. Humans we can see in a range of spectrum that go to the red to the blue, right? But birds, they can expand the spectrum to see from red to blue up to the ultraviolet spectrum. So it's obvious that they see in a different way that we can see, and we need to take this into account in our estimation of coloration of birds. So we use digital photography, and with digital photography, we can estimate, for example, quantify the color in the regular spectrum, but also in the UV spectrum, as you can see in this picture. So we use both the spectrum where we were quantifying the colors in the specimens. So specifically what we did that after all this selection of species, we identify 46 pairs of species. That means 92 species. And for each species, we took one male specimen per species, and we took 6 pictures for each specimen, and we quantified the color, like you are seeing in this video, like selecting the patches on the specimens for 15 body patches. And we extract the colors in the regular spectrum and in the UV spectrum. Using public databases, like for example, AONet, we use 7 morphological traits, measure of 7 morphological traits for about 1,000 individuals. And we quantify for both for morphology and coloration, we quantify how different the pairs of species are using Euclidean distance. So a low Euclidean distance means like they are similar in color or morphology, and higher morphological Euclidean distance means like they are more different in color or phenotype or morphology, sorry. So we were expecting a positive relationship between morphological dissimilarity and elevational overlap, right? And actually, we didn't find any relationship for any of the two clades. So regardless of the elevational overlap, they tend to be very similar in the morphology. The morphological distance tend to be low, with some exceptions. And that could be explained just again by MacArthur. The whole idea of niche partitioning in which, for example, a species can similar ecological similar species can coexist together because they are because they are using different resources of the same locality of the same place. And actually, this has been quantified, for example, using metabarcoding data, in which, yeah, all the warblers in the same locality using different parts of the habitat, and they can consume like proportion of resources different to the other species. So yeah, probably niche partition is explaining the absence of relationship in the morphological data. Now, we were expecting again a positive relationship in color dissimilarity and elevational overlap. And yes, we didn't find any relationship for tyranny, right? Actually, regardless of the elevational overlap, those sister species tend to be very similar in their coloration. We expected the same relationship, positive relationship for OSINs, and actually we found a positive relationship for the OSINs. It was not linear, it was a asymptotic relationship, but still, that showed the relationship that we were expecting. That when species overlap, they have a low elevational overlap. They tend to be like a more similar coloration than when they overlap in the elevation, they are more dissimilar in the coloration. And probably that makes sense in terms of what I was explaining of the habitat the ocean and tyranny occupy, because they are in the canopy, so they can expose their coloration to other individuals. They are more visible to other individuals. So probably the visual communication channel is being important in the evolution of oceans. But this is not important probably in the case of tyranny, in which because they are in those dark environments, so probably they cannot see the coloration of the other species, of the other individuals of the same species. So probably they are using more, for example, acoustic signal in these species. Just to put those results in a broader context, we can see, we can imagine a scenario of the origin of elevational overlap in which, for example, a species can specialize in allopatry, right? But in some points, species can meet together in a secondary contact. And when secondary contact happens, so there are, could appear some ecological interaction that could be in terms of competitive exclusion or character displacement. But what we show here is that these ecological interactions are important in the origin of a, yeah, are also these ecological mechanisms are important in a regional scales, but in our case, in terms of coloration, species recognition, but not morphology. So in the last part of the presentation, so far we have discussed like the importance of ecological factors and the importance of competition in two different scales of times, right? But now we want to explore which are the microevolutionary mechanisms that explain the origin of diversity in tropical mountains. One idea is that mountains are very diverse because speciation rates tend to be high in mountain regions. And actually, some people suggest like higher speciation rates in higher elevation, something that we found in one of the clades, in the tyrannic clades, that there is a positive relationship between diversification rates and elevation. But the whole idea why there is faster diversification rates in higher elevation is because given the topography, the complexity of the topography of mountain regions, of tropical mountain regions, um, there is a stronger differentiation, there is a higher differentiation, a higher population structure of those species, of those populations that inhabit in mountains. And there is the species that have like a higher population differentiation and higher population structure, there is more probability that an speciation event occur. So actually, some works have shown a positive relationship between the population differentiation and the diversification rate. So suggesting that there's a logic link between population structure and diversification or speciation in this case. How species differentiate in mountainous landscapes? One way that you can imagine is, for example, a lineage that has a lowland origin. And what you can also imagine is that this lineage disperse to other elevation in mountains. When they disperse to other areas, this could be considered like a founder effect. And what we know for founder effect is that the population size is going to be lower to those places in which they disperse. So the effective population size is smaller. Because it is smaller, we should have like a stronger effect of genetic drift, right? And the genetic drift will promote differentiation, right? And there is population divergence. But also, selective factors can also act over, like for example, the temperature. Temperature decrease with elevation and is a factor that could be imposed like a harsh environment promoting, for example, local adaptations. And those local adaptations promote also the differentiation to face these new different conditions in different populations. The selective pressures in terms this selective pressure could be in terms of temperature, but also could be in terms of precipitation. Precipitation is also important for different physiological aspects in birds in the tropics. So it's more heterogeneous. Precipitation doesn't change like precipitation doesn't change like temperature is more heterogeneous in the landscape, but it is still very variable in tropical mountains. So the question that we have in this part of the presentation was if the environmental factors are really associated with population differentiation, like temperature and precipitation, and what is the relative contribution of putative neutral variation and putative neutral variation and additive variation, sorry, on such differentiation. An ideal model system to test this idea is, for example, the mountains of Colombia. Colombia is a very diverse country, particularly in birds, and it has like a very heterogeneous topography in which we expect to observe like a very deep population structure and population differentiation in mountainous landscapes in Colombia. And actually, because of that topography, there are also strong environmental gradients that, in terms of temperature and precipitation, but particularly in this window that we were focusing in for this project. So focusing in this window, we sampled 10 species that we consider like representative of the Andean avifauna. And we took tissues samples for about 16 tissues per species in average for a total of 159 localities. That is a total of 175 samples for considering all species together. And we divide our species in three categories: specialists of high elevation, generalist species, and specialists of low elevation. And this is important for something that I'm going to show in a couple slides. So what we did with these tissues is that we generated whole genome resequencing using the Illumina technology. And we did we followed a bioinformatic pipeline that I'm not going to explain in this presentation, but just to mention that in total we got like 63 million of reads for about 24 millions of SNPs for an average of 7x in average for all the species. And the first thing that we did with this genomic information was to explore the spatial patterns of population structure. So we run a mixture analysis and principal component analysis. And not surprising, what we found is that for all species, invariably, we got that there is no population structure. We got a K of 1. And that is not surprising because actually all those species, they have like broad distribution in the Andes. And we were sampling just on a small portion of the distribution. So that means that we are focusing just in microstructure, right? Now, after exploring the population structure, the absence of population structure, we wanted to understand a little well, how is the gene flow in mountains, in those species, in those populations. And you can imagine a generalist species that occupy a broad elevational range. And these generalist species need to cross to disperse through elevational gradient. They need to overcome environmental variables here, right? So thermal bands correspond to barriers they need to cross because probably a lowland population is not adapted to occupy high elevations. Now, imagine a species that is a specialist to high elevations. These, especially of high elevation, they need to occupy these high environments because they are specialized to those environments, and there is a higher disruption of those environments. And this is a higher disruption of those environments. And this is a higher disruption of those environments. And this is a higher disruption of those environments. And this is a higher disruption of those environments. And this is a higher disruption of those environments. And this is a higher disruption of those environments. And this is a higher disruption of those environments. And this is a higher disruption of those environments. And this is a higher disruption of those environments. And this is a higher disruption of those environments. And this is a higher disruption of those environments. And this is a higher disruption of those environments. And this is a higher disruption of those environments. And this is a higher disruption of those environments. And this is a higher disruption of those environments. And this is a higher disruption of those environments. And this is a higher disruption of those environments. And this is a higher disruption peaks of the mountains. So it's difficult for them to move from this point to this point. But in contrast, if you have a specialist of low elevations, what you can find that you can have is like an easier dispersal and gene flow in those species because the topography disruption could be lower. So for that, for exploring gene flow, what we run in this case was a FIMS that essentially is an spatial analysis to estimate migration surface. And with FIMS, we can expect three scenarios. This is one scenario of gene, like homogeneous gene flow. That means there is any individual can disperse to any place and almost mixed scenario, right? There is a case in where there is isolation by distance that is more easy for one individual to disperse to populations that are close, and it's more difficult to disperse to population just because they are far to them, just because it's farther for them, just because of that. But we can have a more heterogeneous scenario in where is more gene flow is more messy here, and that suggests, for example, particular barriers that could be limiting the gene flow among individuals that are very close among them. So with these three scenarios and the three groups of species, we were expecting that, for example, lowland species have, for example, like they follow a model of isolation by distance or a homogeneous model of gene flow, right? Because dispersal should be easier for them. But in the case of generalist species or specialists of high elevation, what we expect is more a barrier model because dispersal is more difficult for them. But actually, what we found is that almost any category of any group of those species can follow any model of dispersal. So, meaning that the absence of population structure is also related with a high heterogeneity of different patterns of gene flow that is not as easy as we were thinking. After that, so we try to explore how population, okay, there is not a structure, but could be population divergence. So the idea is try to understand how environmental factors could explain the population divergence. And for this, we use multimetric regression with randomizations. And first, with this analysis, we estimate the effect, for example, of environmental resistance. That is a metric that involves all the environmental variables in just one metric, using, for example, in this case, the least cost path. And what we found is that from 10 species, 3 show a significant association of genetic distance with the environmental resistance. The relationship could be positive or could be negative, right? Now, we also explore the effect of individual variables. So, for example, isothermality, mean diurnal range, and temperature annual range. And what we found is that from the 10 species, just 2 species show a significant relationship of genomic distance with differences in temperatures, with environmental distance in terms of temperatures. But what was surprising is that 4 of the 10 species show a significant relationship with variables related to precipitation, like precipitation seasonality, precipitation in the warmest quarter, or precipitation of the driest month. Like, for example, Anisognathus, Arremon, Cenops, and Eriomemis, sorry. And this was kind of surprising, and I'm going to explain why. The reason is because when we calculate genomic distance here, we were using the whole genomic variation. That includes neutral variation and adaptive variation. But we can assume that most of the variation is neutral. So why there's an effect of precipitation-related variables to neutral variation, promoting divergence in neutral variation? One explanation is related to how species synchronize the reproduction with precipitation. So species tend to synchronize the peaks of reproduction with the rainfall regimes, right? You can imagine a scenario in which there is a population that reproduces at the beginning of the year, two populations of the same species. And this population, one individual of this population dispersed to another population that they have a reproductive phenology to the end of the year. Because they are asynchronous, this individual cannot disperse to the another population. So there is a kind of temporal isolation between the population. Actually, this is evidence in tropical birds that there is a significant relationship in the precipitation asynchrony with the genetic distance of the species. So maybe explain why we found mostly precipitation variable related with neutral variation, neutral divergence. After that, what we did was try to separate the neutral variation from the putative adaptive variation, right? And for that, and when we did that, we could rerun this analysis, this multimatrix, but separate the neutral of the adaptive variation. For this, we use RDA, that is essentially like a multivariate version of a Manhattan plot. And what you can see is here is like you can put in a multivariate space all the SNPs, all the SNPs in your genome that are represented for these little dots, and all the SNPs in the periphery, in the extremes of the periphery of the hypervolume represent the outliers that are like lossy under selection of like positive selection. And this analysis also is nice because can show you with which environmental variable that differentiated need is associated. And when we rerun the multimetric regressions, the environmental distance with the genomic distance using just the adaptive variation, we see that in the case, for example, of a remon, there was a positive relationship in a variable related with temperature. But this relationship doesn't exist with the neutral variation. So this relationship just with selective variation happen in a remon, but also happen in diglossa, also happen in Metallura, also happen in Icorrina, and also happen in Mionectis. The only species in which precipitation has a significant effect over adaptive variation was in the case of Erioignamis. So yeah, looks like temperature is actually driving the selective variation and promoting population differentiation, but driving selective variation in those species. So just to conclude this part of the talk, so what we found is that probably precipitation is mainly explained the overall genetic variation that including neutral and adaptive variation. But not just, but mostly, temperature-related variables explain most of the selective variation in lowlands and highland species. This work show how stochastic and non-stochastic process may work in concert, driving population structure, and also how microevolutionary mechanisms can explain the origin of tropical diversity in mountains. So what we wanted with this is like trying to make a connection and a link between temporal and spatial scales. And what we learned was this, that taken together, microevolutionary processes are like coupled with environmental attributes of the mountainous landscape, helping to generate the high diversity mountains, but that competitive interactions inferred by the phenotypes are important in the maintenance of that diversity at different scales in the ecological time scales and evolutionary time scales. With that, I want to finish like just, thank you to all people of my lab, to all the people in the department of the Ontology department of the American Museum, all my friends and colleagues from CUNY and the American Museum too, and the funding institution that support me during my PhD, and the museums that were very important during the collecting the data, schemes for measure the colors and also the tissues. Ready for questions. Thank you very much.
All right, thank you so much, Elkin, for a really fantastic seminar. I hope everyone really enjoyed that. Just some fantastic storytelling and results. So really appreciated it. And I think I speak for everyone as we're receiving many, many claps here. So I'll look in the chat and the Q&A box, and we have Daniel Shoestack who raised his hand earlier in the presentation. If you have a question, I can unmute. Although that was earlier. But if anyone else has a question, you can ask in any of these different ways, and I'll be checking on them. Um, oh, okay. He says he does. He did not. So, um, okay. So I'll just start out by I was just wondering for the final part, were any variables at all significant in the neutral or genome wide as but not the adaptive? I know some were both, some were just adaptive. Were there any that were not adaptive, but yeah.
Yes, there were, I mean, we have like different, when, no, when using adaptive and neutral variation, there were just very few cases. I remember like two cases. I think remember an Icorina and another case, like,
Hmm.
yeah, an Icorina. And I think just an Icorina show like a significant relationship of variables in terms of temperature with the neutral variation. Uh, so yeah, just in that case. So
Yeah, I was just wondering if the significant relationship with the neutral variation always accompanied a significant relationship with the selection variation, or if there was anything that was only significant for neutral variation, then wound up not being significant for selection.
I I think it wasn't the case.
Okay.
I think it wasn't the case. I mean,
Mhm.
what you're assuming is that in the previous result like this,
Mhm.
in those cases, yeah, in those cases, for example,
Mhm.
uh, this is like mostly neutral variation because you are using the whole genome here. So yeah, in these cases, probably in these cases, I don't exactly remember here, but I think in some cases, a couple cases, they could be significant for adaptive variation, but I think most of the cases were just significant for just the neutral variation,
Oh,
for
okay.
including
Mm.
those cases and
Mm.
those cases. Yeah,
Okay.
using the whole
Gotcha.
genome. Yeah.
Gotcha. Um, and so the selection includes the coding regions. Is there any way to test the relationship with like promoter regions or anything that would influence expression?
We we don't know that yet. Actually, we are studying the like in the gene ontology, trying to detect what corresponds to actually genes that codifying for something. And actually, wanted to bring to this presentation, but I wasn't. I didn't have time to include that here. We have some percentage of SNP that are located in codifying regions, but the percentage is very low. What we want to know is understand which genes are
Hmm.
those outliers SNPs associated. But yeah, we don't know that yet. That is the
Okay, cool.
last step of this project. Yeah.
Okay, cool.
Mhm.
It would be interesting if some SNPs correlated with some
Yeah.
variables, precipitation, and others with temperature or something. Very cool. Um, what else do I have here? Yeah, I also wanted to ask a naive question. I apologize if I'm missing anyone. I don't believe I am. About one of the early parts. Can you so sometimes they're not at equilibrium at a group, say tanagers or whatever, are not at equilibrium at a certain elevation. This was from the first part of your talk. Uh,
Yeah.
but can the analysis be done to tell that they're like at equilibrium at a certain altitude and then they are at equilibrium at a different altitude? If they're like like still or not at equilibrium at a low altitude, is there a way to conclude that that group is at equilibrium at a high altitude and then you can sort of determine where on the altitude they're no longer at equilibrium? Is that even a possible thing or does that even make sense?
Um, uh, are you seeing my screen? The screen?
Yeah, yeah.
Yeah.
Hmm.
Okay. Um, I mean, I think that the conclusion of equilibrium and equilibrium is given by the overall. by the overall this trend in the elevational gradient.
Okay.
Sorry.
So I think the answer is to my question is
Uh,
no.
yeah,
Um,
yeah,
yeah.
because you need to. Sorry, my cell phone is. Uh, yeah, there is not because you need to understand what is happening in the other communities in other part of the elevation in the framework that we are
Okay.
using. Yeah.
Okay, got you. And I'm also gonna ask, you selected one male for the color analysis portion. And obviously, that's a huge amount of work, but I'm wondering, was there any de decision making that went into selecting that male specimen?
The argument here is that we assume that sexual selection is mainly over the male in most of the in most of the species, at least in the species that we are studying. That if they are dimorphic, there are some cases that they are dimorphic, but most of them they are monomorphic. So that was the decision was based on that. Um, so yeah,
Got you. I was wondering about that individual male in particular, but uh,
yeah,
we only have a few minutes and some other people are asking questions.
okay,
So
yeah, yeah, yeah.
I don't know if there's now all the questions are coming in, but we only have a little bit of time. So okay, so let's go ahead. I don't want to take up all the questions for myself here. So Quinn McCallum asks, phenomenal work. I was wondering about your population structure results in the third part of the study. I know you inferred an optimal K of 1 for all the species with admixture, but your migration surface results show that barriers are important for some species. Did these species also show discrete structure at higher values of K using admixture? And did these conform to the migration surface plots? Uh, did there seem to be a trend in terms of which species showed barriers in the migration surfaces? For example, were they understory species or have a low wing index? If you can remember those questions, Elkin.
Yeah, no, no. For those species that we have like a barrier model, like more heterogeneous gene flow, there is at least with those analyses, we were not able to detect a structure like deep structure. Probably there are other kind of analysis. I remember the name of one that is something like fine structure or something like that. That is for probably for this case like microstructure. But no, I mean, I think that is the advantage of this migration surface analysis that you can, even if you cannot see, because also could be like an effect of sample size, could be, I'm not sure, because we are using in areas like 16, so could be another explanation. But it's just that probably if you see the distance of those population is about 300 kilometers. It's not that probably enough to detect something very deep. And the other explanation is that because we are focusing in this area, we try to do this on purpose. There is not like a deep physical barrier here, like a deep valley or something that like is like limiting the dispersion of individuals. Is this what we have here is the continuous gradient in the intercordillera. So it's just almost just the effect of environment here, the elevation, topography, environment, but not the effect of a particular population, like phylogenetic break that you have for a deep structure that exists for most of the species. Actually, there are paleogeographic studies that show that there is a strong population structure in some of them, but not in this case. The another question was, yeah, about, for example, traits that could explain the absence of a structure. And yes, I think that could be another explanation that, for example, Hanwin index, the heterogeneity in the morphology and the ecology, that some of them they can disperse more than another, even they are especially for high elevation or especially for low elevation, this kind of ecological, morphological and ecological factor, like how if they are more propense to go to another places and explore more area, or they are more, like that they don't move from the place that they are born. So I think these kind of factors could explain the absent, I mean, the mix of result in gene flow. Yeah, I totally agree with that.
Okay, and as long as you're okay, Elgin, we'll run a few minutes over here
Yeah,
just for the last
yeah,
few
sure.
questions. Anna asks, in the results about the positive relationship between overlap and color disparity, how do you explain the asymptote at the end of the relationship? So we might have to, yeah, see that.
Yeah. Here. So yeah,
Yeah.
these are the 4 points that generate the dispersion, the dispersion in the relationship. Actually, yeah, we have a modest relationship with 18, with the explained variance is about 18. And that is produced for those 4 points. And here I remember that at least one of those are, for example, some icteridae, some Sarcoides, I think. Um, Probably one reason uh, is that they can coexist together in elevation, even if they are similar in morphology, but probably because they are diverging in different in another communication channel, more, for example, more in the acoustic channel. So probably they don't diverge in morphology, they don't diverge in coloration, but because of that, probably they are diverging in their songs. Because if a species recognition is important in oceans, because the coloration, so it could be also possible that species recognition is important in the acoustic channel. So that could be another mechanism for which they look similar yeah, they look similar in the plumage, they are similar in the morphology, but probably they are using different vocalizations in the same place, and that's okay for them. Yeah. So that could be a potential explanation.
And we had one more question. I think Esteban, you removed the question maybe, or I don't see it anymore. But I think that Anna's question leads very well into what Esteban's question was,
Okay.
which was asking about other variables that you hadn't tested yet. So you just brought up song, and I can't remember exactly what Esteban wrote about the variables to test, but I guess are there other ecological variables that what would you look at next?
Sorry, the question was, uh,
Um, I'm trying to remember it. But I think it was basically you tested some ecological variables. Obviously, you can't test all of them.
yeah.
Are there others that come to mind that you would look at next?
Um, no, I think what you
For example, he said nesting type was one of the things he had said.
yeah, yeah. I actually I wasn't thinking in that.
Mhm.
Mm, I don't know. I'm sure for some species, for example, the reproduction site, the places that use the reproduction to put nets, could be for sure important in competition. For example, in woodpeckers, the holes in the trees is a limiting factor for them. But I'm probably, yeah, I'm probably for other species that have very specialized architecture in the nest. Uh, will be important in competition. Not sure. I was thinking something, yeah, for maybe it's important. I don't know in an insignificant way, but I will expect something more that at least I would like to explore more is in the acoustic factor. That is something that I want to include here. Like actually
Mm.
measure if the acoustic signal, because there are many examples of playbacks in which they respond very aggressively to a song that is very similar of an individual that is, a neighbor. So that suggests that vocalization is important for example, delimiting territory. So for sure, that will be important in that for yeah, measuring.
Okay, so we are 5 minutes over here. Ramona, are you still there? I just want to make sure, Ramona, who's
I'm still here. Yep, I'm still
you
here, and I think we have the room until 1:15.
okay, all right. Well, Elgin, if you're okay with
I'm
it,
okay. Yeah.
okay,
Hmm.
we'll just we'll keep going here. So uh I will unmute Juan here. Uh Juan Ramirez, you can I believe you can speak now. Um but while we're waiting,
Hmm.
Esteban wrote maybe a version of the same of his question from before. Are there any adaptations of natural history uh, at the species levels uh, in nesting biology and migration? So if you feel like you touched on that already, that's okay. But um,
How's the question again?
oh wait, now Esteban is raising his hand, so I'm gonna let him talk since I'm not I'm he'll do a better job of
Hi.
explaining.
Hi,
Uh,
can you hear me?
yes.
Okay. The question was, I don't know how to put it in a right way. Is there any adaptations at the species level, like the site of nesting, and is there any niche partitioning in the place where you can as a bird nest in, for example, in all of this gradient of habitats, because one could expect that is there there is a habitat disparity for picking the nesting site for the species, and also this could have been driven by a migration and yes, and capacity of the capacity uh, of
Dispersion.
migration, like the wingspan or other things that are indicated indicating of of que son indicativas para la para para saber en dónde estás, para saber en dónde anidar y en
Okay.
dónde vivir en este caso.
Uh, I think
Sorry for saying in Spanish
Oh
for
yeah,
no,
no
it's okay.
problem. No problem.
It's
people.
Yeah,
okay.
we just wanted you
Uh,
to ask a question. Yeah,
mhm.
appropriate. Mhm.
Um, for sure, there are probably in very more specific ecological trait, like for example, I will consider a specific trait, like an ecological trait, like the side of reproduction, or the nesting side, like an important factor. And I think they are competing in that there is a niche partitioning for species locally, and that they compete for the niche places. But I don't think, uh, I don't know if that is enough. Maybe that this is enough to, for example, limiting the coexistence of two species. I haven't read anything related with that, but yeah, I think it's possible. But yeah, something that is you have to measure like more locally, and this kind of scales, for example, in a regional scales or crucial scales, is difficult to test, but for sure is possible.
Mhm.
Also dispersion, the dispersal is important. Not sure migration. In this case, no migration because we don't use migratory birds, but still could be, yeah. We didn't use like migratory birds,
No, no,
but
no, but sorry,
locally, yeah, like dispersion from one place to
locally,
another. Yeah.
but in a altitudinal
Oh,
gradient
okay. No,
as
yes,
well.
no. In that sense, yeah, I was thinking in something like that. Like, for example, in hummingbirds, they perform like elevational migration, and that probably when they are there are some species of hummingbird that stay in the same place where there is a dry season, for example. But others go to another place and probably following resources. And that is a way to reduce competition. So yeah, I will agree with that.
Okay. Thank you.
All right, and Chris Witt has a great question here about those those data points that are on the slide here. Actually, do you think plumage mimicry could be an alternative way to explain high plumage similarity with high elevational overlap?
Plumage mimicry. Yes, but I don't know. I'm thinking if we can define similarity between sister species like mimicry. I will think that mimicry will be in a species that are not they are closely related, but not necessarily the sister species. So in that case, I will not think like because we are using sister species. Also, we are using sister species. So in that case, I will not consider like mimicry like a scenario to explain the dispersion. If they are similar, it's because they share a common ancestor, but not because they are evolving like mimicry. But not sure if I'm understanding the question properly.
Yeah, I think I understand that. I also, well, I don't know, but I'm now remembering that these are all sister species as well. That answer makes sense given that relationship. But uh, I'm just checking. I think I got to everybody's questions. I am sorry if I didn't, if I missed anyone. Um, yeah, and Chris responds by saying, it's a fair answer, but it's not I don't think it's possible to rule
Okay.
out mimicry between sister
Okay.
species, which which is fair. It would be interesting to
Okay.
know if there's tools that can be used to look at that.
Yes.
But we're gonna get I think we're gonna get kicked out in like 1 to 2 minutes. So I want to thank everybody for being here and supporting Elkin's seminar here. Again, a fantastic seminar. I would see it in the PhD dissertation version as well. Uh, and I want to thank everybody for being here with us for this semester, for all these seminar series, and because of the support of the AOS community, we will be back again in the fall. So I really appreciate everyone being here. What a great way to end it here today. And I will see you guys in the fall. Thank you so
Okay.
much, Elkin.
Thank you.